one of the bigger ones that we can kind of point out on this is prevent overstimulation of our immune system. Do you guys remember some of the things that we talked about, particularly the disease processes that attacked our endothelial cells, right? The ones that attacked our endothelial cells and created vascular leakage was because of that extra cytokine causing that overstimulation of the immune system, causing vascular damage. So some of this can be prevented by some vaccinations. That cytokine storm that's created by the overstimulation causes a lot of that vascular leakage that we talked about during IV. So these are some of the things that immunizations can do for us. This looks a little bit crazy, right? Pretty busy, it's like all over the place. Okay, but I kind of sort of want you to understand how immunizations come about. It isn't one agency alone. Okay? In some cases, it's driven by money, right? So the NIH is the National Institute of Health. They usually work with industry. Okay? They work with all the companies, all the pharmaceutical companies to be specific, right? And they often support and or conduct early research for vaccines that we need or improve vaccines that we've already had out. So NIH works with civilian companies to develop that stuff. Once it's been developed, it goes to the FDA. The FDA reviews the clinical trial data from the NIH. Okay? No vaccines in the U.S. can move forward without FDA approval or authorization. So if you remember when we talked about IV, some of the things that we talked about, we said FDA approved, not FDA approved. This is one of the reasons why. As we go through the immunization policy, the way it works is it has to go through an FDA, and it needs their approval to continue to move forward. As it moves forward, it goes to the ACIP, which is the Advisory Committee on Immunization Practices, and they review all the safety and efficacy data, right? Post-approval from the FDA. And once they look at that, they issue clinical guidelines. And those clinical guidelines are generally based on age, right? Risk factors, and they're the ones that come up with the dose and scheduling to give people. After they do that, then it goes to the CDC. Okay. CDC are the ones that do the policy adoption and distribute the guidance. Right. It's all clinicians, public health, and all the other places, right? So if you come, right, so you're with the civilian folks and the pharmaceutical companies, they review all the work that they've done. Once they approve it, they look at everything that's been approved and come up with guidelines. And that stuff goes to the CDC. These guys make policy. And then we, as federal providers, can get it from two different ways. Okay? We can get it from the CDC straight, or it can go to DOD, which means it goes to our surgeons, respective, our services. They review it and then they can push it down. Usually there's no changes when we're stateside. The changes do become slightly different when we deploy or we go overseas. So those are the two areas that end up coming to us as DoD healthcare providers. Right? Once we get that information, then we have to assimilate it, and that is what we have to partake to our patients, to our patient population. That's how the whole entire immunization process works. So immunization immunology, the immune system's main purpose is to protect you from foreign domestic invaders. So stuff that may be on your skin or stuff that actually gets into your body. Right? That'll be all those lymphocytes, T cells, B cells, monocytes, macrophages, all those things, right? And here it also gives you that big one at the bottom here. That avoid abnormal or exaggerated immune responses by hypersensitivity reactions. So overall, there's 4 types of hypersensitivity reactions. There's the type 1. Type 1 is usually the immediate one. The anaphylactic one usually happens right after you do something that aggravates you, right? There's a type 2, which is a cytotoxic one. Okay? And that one is basically your immune system destroying your cells that are involved. You have type 3, which is the immune complex or mediated hypersensitivity. And that one is the common one that can be associated with the endothelial cells being attacked, right? During some of those IgE diseases that we talked about. And then type 4 is a delayed type, which is cell-mediated. It's another hypersensitivity, which can also fall under some of the issues that we talked about in some of the disease processes in IgE. So here's some definitions you probably need to know. Immunity, the ability to tolerate the presence of material indigenous in the body, and to eliminate foreign material. All that really means is your body got something, your body realized you had something, your body developed something against it, i.e. antibodies, okay? And those antibodies will remain in the body, i.e. usually memory B T cells, so that if you do get it again, you can fight it, right? Then you have your antigens, which are live or inactive substances, generally proteins, which produce an immune response. Those are the things that you get, right? That's the substance that the immune system recognizes as foreign, not right, so it's going to develop something against it. The antigens are usually the trigger your body to develop antibodies and respond against the problem. Because the next one are antibodies. Protein molecules produced by the lymphocytes that fight antigens. The antibodies are going to fight those antigens that are causing you a problem. That's what they do. They identify the problem and they neutralize the problem. They don't put it in military verbiage. They fight viruses, bacteria, toxins, all that other good stuff. And then vaccines are a product that contains antigens, right? So those antigens can be in a whole bunch of different forms. They can be live, they can be dead attenuated. Okay? All they're doing is stimulating your body to notice there's something foreign so it can develop those antibodies and develop the memory T cells so that you can fight without actually causing you the disease. And therein lies the big difference with the vaccines. Not causing you the disease. Which factors in implicating or impacting vaccine effectiveness. And we talked about some of this stuff as we've gone through IE as well, right? Age is a huge differentiator. If you have comorbid conditions, that makes a huge difference on whether you should or shouldn't get vaccines. For the most part, you should. Okay? Prior exposures, right? When we talk about dengue, you don't really give vaccines unless they've been prior exposed to it. Right? They're seropositive. Then you have other vaccine characteristics, right? The mode of delivery, you need to figure out which way you're going to give it to. Right? Is it going to be IM? Is it going to be sub-Q? Is it going to be intradermal? Is it going to be oral? Is it going to be intranasal? A lot of different ways to give these things. Is it a live vaccine or is it an inactivated vaccine? Right? Because then we know who we can give it to, right? Because we don't give live vaccines to pregnant, right? Or severely immunocompromised folks. Here we've got vaccine composition. We'll talk a little bit about that here in a bit. Here it talks about adjuvants. Those adjuvants are just substances that enhance the body's reaction to help the response to be stronger with using less of the antigen in your body. Okay? The most common adjuvant that we use is aluminum. And that matching to circulating strains is a hard one, right? That one we could look at influenza. You have to figure out next season what is going to be the most common, or what's going to be the hardest hitting strains of people. And that's what we develop next season's influenza on. Sometimes they get it right, sometimes they don't get it right. And that goes back to that antigenic drift, antigenic shift. You guys remember that from ID. That stuff is common for influenza and COVID nowadays. Okay. Here are some type of immune responses that you're going to get. So immunologic defense here, you have your innate system. Okay. Your innate system is the first line of defense. Okay. That one's rapid and general. Real clear. It's got multiple things. It's got a physical barrier, right? That part of that physical barrier would be your skin, right? This membrane. And then after that, you have your cells. You have your macrophages, your dendritic cells, your neutrophils. Those are the ones that get activated for that. Then you have your high specific, your adaptive system. So the adaptive immunity is a lot more specific. Those are the ones that develop after the exposure. Those are slower for the initial response. These are the ones that create your memory cells, okay? Your MPH B cells. This is the specific IgGs later on. And you have two aspects to that. You have that cell-mediated, which is basically your helper T cells and your cytotoxic B cells. Those are the guys that go and destroy all the infection for the most part. And then you have your humoral aspect of the adaptive system. And those guys control the B cells and your antibodies. Now, if you want to put some of this stuff in military terms, we'll say cell-mediated is kind of like little snipers. They are extracellular. So anything outside the cell, your cell-mediated cells are going to go ahead and try to kill you. Okay. Anything that's or anything that's intracellular goes with cell-mediated. So anything inside the cell will try to, those antibodies, T cells will try to kill. The humoral is extracellular, so anything outside the body or outside the cell, those cells will try to kill before they get inside the cell. So humoral, extracellular, cell-mediated, intracellular. And there's two different locations where they kill whatever the invading or offending pathogen is. And here's some more definitions for you. Your active immunity develops antibodies and responses. Okay. This is usually after exposure to antigens, the infection or vaccination. This is the one that leads to memory cells. And the next one's passive. Remember, we talked about passive. Here it's pretty, the example is shown is maternal antibodies given to the baby. Okay, not a problem. The baby will receive them, but when the baby comes out, the baby will only be protected in generally up to 6 months or so status post-delivery. Okay. But that passive immunity to the baby does not confer memory cells. Okay. So even though they were protected for those first 6 months from the mom stuff, they over time have to start developing their own. Okay. So passive stuff does not develop antibodies or memory cells. And then you have your herd immunity protection, because those around you have antibodies or response cells. In most time periods prior to right now, right, we had a large amount of people that were completely vaccinated, right? Nowadays, a lot more people are starting to decrease that vaccination, which also decreases our ability to do herd immunity, right? So we'll say we can take this whole entire side of the room right here, right? Right now, let's say everyone is vaccinated, okay? Except for that guy right there with glasses on. All right. If I'm a disease process, I have to get to him, but I can't because I can't go to him, I can't go to her, and I can't go to this whole first row, right? So I can't get to him. Now, what's happening nowadays? People are choosing not to be vaccinated. Okay? We can use measles as the example. Okay? We will say this whole first row here chose not to get vaccinated, and those two right there chose not to get vaccinated in the second row. The guy with the glasses is still the guy that cannot receive the vaccine, so he is unvaccinated. So now that I am a disease, say measles, I can get into this guy in the whole front row, and then I can get into the next two people in the next row, which makes him susceptible to getting the measles now, because of the lack of herd immunity that we're starting to see now. Okay. So understand that as we continue to grow, with more and more people that do not want to get immunized, our ability to do herd immunity is decreasing drastically. All right. Here's your herd immunity, indirect protection from infectious diseases for those not immune. It's pretty simple. So, hit herd immunity threshold is a percentage of population that must be immunized through vaccinations or actually naturally infected to a specific disease in order to stop the spread within the community. Right? In this case, this was the entire community, this side of the room. Everybody was, except for one person. Not a problem. Now we go to basic reproduction number. That R sub-zero there is what we call R naught. R naught. And that's just a measure that's used by epidemiology. Okay. So, and it describes how contagious an infectious disease is. And it describes how infectious the disease is. And generally, it's just the average number of people that one person can infect. So what that is. Or number that he can fully infect within a susceptible population, which means none of these people on this side of the room are immunized. One person gets sick, how many other people can he infect that are not immunized in that group? That'll give us that R naught. So average number of new cases caused by a susceptible within a well-mixed population. Literally all that means. Okay. There's some types of vaccines. This is not an all-encompassing list. These are just some of the vaccines. You have live attenuated, you have killed organisms, you have toxoid, conjugated subunits, and mRNAs. Okay. So your live attenuated are weakened strains or weakened versions of some of those pathogens. So, because those are live, there is, in some cases, a very, very small possibility that you can give it to someone. It is extremely rare. The last time I looked for oral polio numbers, I think it was 1 for every 2.7 million people that it may have caused polio. Numbers are pretty huge. That's why they say it's very rare that any live stuff can cause the actual infection. Okay. killed organism is the inactivated version of that. Alright, so the pathogen itself is completely inactivated. Alright, it's been killed by heat or chemicals, whichever they chose to do, but the pathogen that they're actually giving you in the inactivated is actually dead. So the only thing it's doing is the outer proteins are actually stimulating the body to develop those antibodies. Nothing different. You have your toxoid, okay, that's also inactivated bacterial toxin rather than the organism itself. You have those conjugated subunits, those are weak antigens. They're just weak antigens. Usually made from sugar molecules with appropriate protein carriers that take them into your body that actually mimic and stimulate those antibody productions. And then ultimately here you have your mRNA vaccines. Those are synthetic messenger RNA vaccines. Okay? So it's a synthetic molecule that goes in there. According to the studies, they say it's harmless. Okay? And the immune system reacts to that protein, and that protein is what stimulates the individual person to build immunity. Okay? Most folks had never heard of mRNA until COVID started. mRNA vaccines have been studied for well over 20 years. We just had not put them into practice because we've been doing research in the background to see what kind of effects we would get by using that. It just so happens that we really needed something during that COVID time period, and this was something that they could develop really, really quickly, specifically against the one particular targeted COVID issue. Okay? So that's why those things came out. But like I said, this is not an all-encompassing list. There's a whole bunch of other kinds. There's recombinant protein vaccines. There's virus-like particle vaccines. There's recombinant subunit vaccines. There's a whole bunch of different ones. This is like its own study on its own. So, you know, we're only talking about some of the main ones that you guys are going to be dealing with. All right. So here we're going to talk about vaccine adjuvants. And this is just a substance that's added to a vaccine that enhances the body's immune response to the antigen. Right? Part of that vaccine kind of mimics the disease organism, but the adjuvant stimulates it even more. Okay? So the adjuvant itself does not act like the antigen. Okay? It just helps the immune system respond more strongly and more effectively to that antigen. And that adjuvant is added in there because that will allow us to decrease the amount of antigen that we put into that vaccine. And one of the more common ones that we use in the US, it says there, is aluminum. Okay? Because it stimulates immune cell response. What it does. And it promotes antibody production. Questions always come back as, are adjuvants safe? The answer is yes. According to all the studies that we currently have. Okay? Right now, the way it stands, according to all the studies we have, adjuvants have undergone extensive testing, and none of the testing has shown it to be detrimental or harmful. Remember, we do have some aluminum in our bodies, and our urinary system usually gets rid of it pretty quickly. Now, some of this aluminum stuff may cause redness or soreness at the site of injection. That might be one of the reasons why some vaccines cause you redness and soreness at the site of injection. Residual matter. So this is dependent on what it's grown in. Vaccines are grown in specific mediums. some are antibiotic mediums, some are egg protein mediums, some are gelatin mediums. There's a whole bunch of different mediums that these things are grown in. Right? And what this thing right here is telling you is that there may be trace substances of this residual material within your vaccines. Okay, these are not active ingredients, and they typically present in very small, non-harmful amounts. Right? This is what they tell us, based against whatever is grown in. Vaccine preservatives and stabilizers. So, in the next slide we'll talk a little bit more, but they use preservatives and stabilizers in vaccines so that they don't generally go bad. More so for the multi-vial doses. Nowadays, any single dose vial or injection generally does not have preservatives or stabilizers in it. Usually, it's the multi-dose vials. Right? This helps maintain potency, right? Because as soon as you open up that multi-dose vial and you draw out the first one, you have a limited amount of time to use it. Anything you're introducing into it can contaminate it if you're not doing it appropriately, right? So you're using some of those preservatives and stabilizers to make it last longer. Right? You want to prevent bacteria or fungi from getting in there once you open it up. That's what they're supposed to be used for. Right? They keep the vaccine safe and effective after we open it, and we keep it stored away until we need it again. Right? Most vaccines nowadays usually do not contain preservatives, especially if they're packaged as single doses or pre-filled syringes. The big one that we always talk about when we talk about this stuff is thimerosal. Right? That's the one that folks keep saying causes autism. Right? Now, back in the day, almost all vaccines had it. Nowadays, it's very, very minimal, and none of the childhood vaccines have thimerosal in it anymore. Thimerosal breaks down into ethylmercury and thiosalicylate, which are both cleared pretty quickly from the body through the urinary system. Okay? And then, as an adjunct with the ethylmercury, it also gets pooped out. So, two different means to get rid of the ethylmercury, and usually thiosalicylate is usually more so urinary. Okay, common concerns about vaccines. This is where we can talk about some other stuff. Okay? Specifically, thimerosal. One of the big things that we're going to have to discuss and educate your patients on are about the one big article that came out in 1998 that was written in the Lancet by Dr. Andrew Wakefield, stating that he found a link between those immunizations with thimerosal and autism. That's what it came down to. But there is no scientific evidence that supports that. Extensive research since that has been disproven. Okay? Here's the thing where we have to remember what we learned in freshman semester, right? Mr. Master's thesis stuff, where it said, hey, you have to be able to read the research and see if it is a good research document or not. This was not a good research document. Right? Any reason why it was not a good research document? You guys remember what the N number is supposed to be? One. No, N number is supposed to be the number of population that you have in your study, right? That includes your control group, right? So do you want a middle number or do you want a big number?
Big.
You want a big number, right? Okay. Anybody want to guess what the number was for this gentleman's study? How many? 200. Higher or lower?
Lower.
Okay, what do you think?
100.
100. Higher or lower? Lower. What do you think? 80. Higher or lower? Lower. What do you think?
60.
60. 40. Higher or lower? Lower.
25.
How many?
25.
25. Higher or lower? Lower. 18. Higher or lower?
7.
Okay, it's more than one. Okay, it's more than some. He had 12 people in his study. 12. Do you think he had any control group?
Control group of 1.
He had no control group. There was nothing compared to this. Do you think he had specific processes to do certain things with patients so that he can compare whether it was good or not good?
No.
No. I'm gonna just tell you guys, if you guys want, you guys can go and look at it and see all the stuff that happened. But, right, N of 12, no control group. What else do you think is really bad when people don't say stuff about their affiliation with certain things? Say again?
Usually it's because they have some kind of loyalty to them, they got paid by them.
Okay, so that should have been something that you guys discussed when you guys did freshman master's thesis. Right? Hey, people should disclose who they are associated with or getting paid by when you're doing that research. Who do you think was paying this guy to do that research? I will tell you it's not the pharmaceutical company. Right? So during that time period,
Wellness influencers.
real influences during that time period. Right? So during that time period, there were a whole bunch of lawyers that were trying to sue the pharmaceutical companies for some of the vaccines that they were coming out with. Right? Understand that during that time period we had a whole bunch of new ones, and because they weren't doing thorough research and they were releasing them, they were having some serious issues with some of the vaccines. Okay? So there was a group of lawyers that paid this gentleman $700,000 to do this research so that they could use it in court against the pharmaceutical company. Do you think he mentioned any of that in his study? No. Okay. One other piece of information that he also did not mention, he himself had patented a new measles vaccine. And part of the autism piece was MMR. Right? He didn't say he had patented a brand new measles vaccine. Right? So you start showing a big pattern of a whole bunch of things happening, which is not good for you. Not to mention your study, not good study, right? So that stuff happened in 1998. Right? In 2004, 10 of the 12 co-authors retracted their paper interpretation. Okay, we don't agree with this stuff. We don't. Now, this guy is not from the United States. This guy was from the UK. Okay? In the UK, they have what they call a General Medical Council. That General Medical Council is the council that oversees all the doctors and gives them their licensure and all that other good stuff. Okay. He had a trial in 2010 where he was found guilty. At that point, the General Medical Council in the UK pulled his medical license. No longer doctor. In 2010, the Lancet that printed that study also put out a retraction against that study. So it took from 1998 to 2010, 12 years, where this stuff was out there. And people were looking at stuff and trying to associate these things. Right? But since then, there's been well over a dozen studies, high quality studies that actually meet the appropriate standard, right, of what a study should be, demonstrating and using millions of children, okay, and showing no link between vaccinations and autism, right? Now, with that being said, there was a huge outcry in that interim, right? Not knowing that the study was not good, took them 12 years to prove it and to put it out. So naturally, the pharmaceutical companies reacted to that and removed thimerosal from childhood vaccines. Do you think autism rates have increased or decreased since 2001 when they removed thimerosal from childhood vaccines?
Increases.
So it's increased. And autism has increased since then. All right? So if we're trying to do relational cause and effect, right now we don't have it. Okay? We don't have it. So just keep that in mind. Here it also tells you natural immunity is better. And if you want to go through natural immunity and you want to get some of those diseases, knock yourself out. Remember, you've got some complications that expose some of these diseases. You may end up in the hospital with some of these diseases, and you may end up dying from some of these diseases. So you can prevent it. Okay? To me, vaccines are a much better thought process than actually going to get a disease. The other piece for me to use, right, is social media is not your friend when you're a medical provider, right? I don't know where X went to medical school. I don't know where Instagram went to medical school, but that's where some people are getting a lot of their medical information from, and that is what they will bring to you. You will have to educate them appropriately on this stuff. Okay? Specifically vaccinations and the importance of, and the studies we have shown. Because you're going to have to dispel all the information that they're getting from their social media feeds. The other big one that you're going to have to fight is a distrust of pharmaceutical companies. And I don't blame them. I wholeheartedly agree. Okay? But what we have to do as providers is just give them the best information that we have available so that they can make the best decision that they can. Okay? And we, as providers, have to do that without bias. All right? We have to do it without bias. And that sometimes is kind of hard. Okay? All right? This methylmercury is a completely different story. All right? This methylmercury is usually what you find in infected, generally fish. Right? It says it's lipid soluble. The problem with things being lipid soluble is they can cross the blood-brain barrier. In this case, it can get to your brain. Okay? And methylmercury is neurotoxic. Now, we put this on here because we need to make sure that we know the difference between ethyl mercury and methyl mercury. Two different substances, okay, where people kind of sort of get confused. Okay. Remember, thimerosal breaks down into ethyl mercury and thiosalicylate. Methyl mercury does not. Okay, that accumulates in your tissue. So they are clinically different chemicals. Now, here at the bottom, it gives you a great example of methyl mercury poison. All right. So there was a company, a chemical company called Chiso Corporation, back in 1932 in Japan, and they dumped a whole bunch of methyl mercury into the Minamata Bay. And they dumped it in here for, how many years is that? 36 years? They dumped that stuff in there for 36 years. With the okay from the government over there. And they didn't start noticing anything till the mid-1950s. So they started in 32 to the 50s. That puts you at what? 28 years? 28 years later, they finally started noticing some effects of the methyl mercury. Okay? And what happened? The methyl mercury affected the water. It affected the sediment, the bottom of the water. It affected fish, specifically shellfish. Right? Now, this town lived off the sea. Right? The Japanese diet really contains a lot of fish. Right? So these folks started getting sick. This right here is one of the worst industrial pollution disasters in history. That mercury bioaccumulated in those fish and shellfish. And as they were catching fish and shellfish, they were eating. It's a major food source for those people. Over time, he started realizing that they were getting mercury poisoning. Okay? People developed a wide range of neurologic problems, physical symptoms that led to permanent or fatal medical issues. I think like 2,300 people died from this. There's more than tens of thousands of people that have residual effect. Right? So I told you ethyl mercury gets put out of the body pretty quickly. Okay? Ethyl mercury and thiosalicylate usually get peed out or pooped out of the body within 3 to 7 minutes. Methyl mercury accumulates in your tissue and it generally does not go away. So the more you eat, the worse you get. Right? Now, some women got pregnant. What do you think happened to the fetus?
Birth defects.
Right? There was a lot of congenital defects. They either had fetal abortions, spontaneous abortions, but for the ones that did survive, some serious congenital neurologic effects. It was bad. It wasn't until 2004 that the Japanese Supreme Court ruled that the government and the Chiso Corporation were responsible for this. How many years is that? How many people do you think died in that interim? That's a lot. It's bad. They ended up having to pay for all these people's medical and everything that went along with it. That was a lot of money. A lot. But we learned, right? We learned from this. Okay. Here's your ethyl mercury. Okay. It is a component of the thimerosal. As you mentioned, thimerosal will break down into ethyl mercury and thiosalicylate. Okay, there's no evidence that shows that either of those two cause any harm, because generally they are repeatedly dealt within 3 to 7 days, at least according to the studies. Now, thimerosal is used in select vaccines in multi-dose vials, so you really have to be careful and you have to read what the manufacturer says. Now, there are multi-dose vials that do not contain thimerosal or preservatives or stabilizers. You can ask for those and buy those. Those are generally more expensive, but they are out there. So depending upon what you get, is what you're gonna have to look to see whether it does or does not. Now, for the most part, anything that's single or pre-filled, one-time use, usually doesn't happen. So those things you don't have to worry about, okay? The four big ones that have the multi-dose vials that you need to concern yourself with will probably influenza, TB, like big T, little d, that would be more so for boosters, okay? Meningococcal and Japanese encephalitis. Those are generally the four that come in those multi-vials that you really have to check to see if vitamins are already there as per the preservatives and stabilizers. So the CDC puts this out, World Health Organization puts this out. This is just information for anyone who wants it or needs it. They can go to the CDC site, and it's there. It'll be there for you for education purposes. If you need to talk to people about this stuff, okay, the information is there. Right? And here it tells you, due to the increased recognition of vulnerability of children, thimerosal has been removed from most U.S. vaccines given to children since 2001. So it's been gone for quite a while now. All right. Here's a whole bunch of other things that people think they're overwhelming your immune system. Your immune system gets hit with hundreds of thousands of things every day. Okay? Over a total of your 18 years, you're going to get 150 antigens from the amount of vaccines that you're going to get. 150 over 18 years is nothing compared to what you generally have to deal with on a daily basis. Right? So, some of the things that people think, generally not necessarily the case. Right? Vaccines overload the system. Your immune system deals with more than thousands of things every single day. Genetic vaccines were, they try to develop, that basic guidelines try to develop that schedule to give it to them at the best time to protect them. And there it says there is no evidence that children get more infections right after they get immunized. People think that they get sick more, it's just a reaction of the vaccine causing them to get a little bit sick. And people, adults, now have the same concept, right? And it will come to you when you are doing your interview for an influenza vaccine and tell them, I get sick every time I get it, or I get the flu every time I get it. You're not getting the flu, your body's being stimulated similarly to the flu so that you can develop antibodies. That's what's occurring. You don't have the flu. Okay? Your body's developing the same type of symptoms so that your body can develop those antibodies against it. So a lot of this really comes down to education. Okay? Education for your patients. Right? Natural immunity is better. You just mentioned that earlier. Yeah, by all means, knock yourself out. Back in the day, they used to have pox parties, so they used to take little kids to other little kids' homes so that they can spread the pox to themselves. We no longer do those kind of things. Okay. Remember, there are repercussions on some of those things if you choose to actually get it versus just doing your immunity via vaccination. Okay. And there at the bottom, that nice big natural immunity is only better if one survives the illness, right? You don't know what's going to happen. Bigger differences, even with some of the natural immunity, right, depending upon what it is, you may only have it for a couple of months status post, depending upon which disease process you have. Right? Some of the issues with some of the vaccines we have, so you'll have to get them periodically. Right? But some of our vaccines also do indefinite protection. Here's some immunization practices. If you have not given immunizations before, these are the core standards for us in the military, usually the same in the civilian sector, for giving immunizations. Right? The DoD has adopted these 8 standards Okay, the Guide to Military Immunization Practice. And they're real simple. Okay? Immunization availability. I should not make it difficult for you as a patient to go get your immunization. Right? There should not be barriers causing problems for your patients to go get a vaccine. So let's just go ahead and take a break before we actually start. That's what screening is for. That is what that screening form that we usually do for immunizations is for. Okay, we're looking at allergies, we're looking at health status, we're looking at previous adverse events if they have had any to not just the current vaccine, but to any of the other vaccines that you have to be careful with, because that may be a contraindication to giving the vaccine. Right? And you've got your immunization record keeping. And that's the rough one, right? Because if it didn't get documented, it didn't happen, right? If it didn't get documented, it didn't happen. So we all have different systems here, right? The Navy has MERS, the Air Force has ASIMs, the Army has MedPros. The stuff needs to get documented in the appropriate system. Nice thing about ASIMs for the Air Force is that Army guys can put stuff in there and it'll cross over to MedPros, so it works pretty well. So make sure the documentation happens. Okay. Training personnel, we, PAs, medical providers, have to do the 1 hour JKO course every year if we're going to be the ones giving out the influenza vaccine. Okay. So beside that training, sometimes they have other training to talk about vaccinations as a whole. So we can do those as well, but we need to make sure that we maintain our training so that we know what we're doing when we're taking care of immunizations. Adverse events after immunizations. We'll talk about that here in a bit with submission of ours. But this piece, more so for us, because we're going to be the ones that are going to be supervising the immunizations, especially the way we do stuff, right? We don't do it in the clinic, we do it in freaking rodeos, or we do it out of the aid station, right? You need to make sure that you have everything set up that you need if someone goes down. Make sure you have your EpiPens, make sure you have a litter, make sure you have a plan to call 911 so that they can come and get that person if they've gone down. Make sure you have a means of controlling the airway so that they don't, in case they have problems breathing. Right? Make sure you have all the appropriate equipment that you need when you're going to do that. That includes oxygen and a BVM, that mask. Okay? And then your vaccine advocacy to protect the family. That one's a little bit different. Okay? That one is a program with the hospital itself, and they usually develop a plan to get immunizations for anyone who is high risk. So if you have a family member that is high risk, you can talk to the vaccine advocacy folks at the hospital, and they can get you in whenever you need to, whenever new things come out. For example, the newer influenza vaccines, you can be one of the first ones that ensure that you get it as a priority. Okay? That's what that vaccine advocacy is for folks that have high-risk family members. Okay. This is what we call a VIS, Vaccine Information Statement. All right. The purpose is to inform the patient of the vaccine that they're about to take. Okay. Now, it's not just the patient, because sometimes it's the guardian of the patient, right? There's a lot of times it's little kids that are getting these vaccines. So the parents need to be well informed of what they're getting and why they're getting it. All right? And this just discusses the benefits and the risks of the vaccine. Now, you should also be available, or someone should be available to be able to answer any other questions that they have before they get the vaccine. Now, this is required by law. This started off after the National Childhood Vaccine Injury Act back in 1986. led us to the development of this vaccine, this VIS form. This is run by the CDC. It's on all the CDC websites. And people can access them, and they change every single year. Every single year they get updated. Okay. Now there's a couple of different ways you can do this VIS presentation. You can put up a QR code and allow them to use their phone and pull up the VIS on their phone. You can make a giant poster and put it up top as they stand in line before they come and talk to you to get screened. You can print out a whole bunch of them and give everyone an individual form. But everyone needs to have, or at least been able to read this stuff before they come see you for screening, okay, in case they have any questions. Storage and handling, generally we as providers don't deal with this, but if we do, we have to take the cold chain management class, JKO, and that really means that you need to make sure that you follow the appropriate handling of the vaccines from the point of origin to the point of use. In many cases, the most important piece is how to keep it at the appropriate temperature. And it's got to be read every couple of minutes once it's out in the middle of nowhere, because we want to make sure that it doesn't lose its efficacy. So one of the drawbacks that we had when we were using the intranasal live influenza vaccine that they think was causing most of the problem was that we were not able to keep it at the appropriate temperature everywhere we moved it to. And when we held it at a specific location X number of times before we actually gave it to someone. So temperature is really important when we cold stage cold chain management. Indications and contraindications. I think we've kind of covered already, right? Age, it's dependent on the type of vaccine that you're going to use. Pregnancy, we're never going to give them live vaccines unless we talk about risk versus benefit. That generally wouldn't necessarily be us as much as it would be an ID guy or an OBGYN guy. Occupation, based on what you do, that's really going to determine the specific types of vaccinations that we're going to give out. For us, as military folks, I would suggest, particularly the Army guys, if you go with the MedPros, you can do a drop down of an AOR, right? Or you're going CENTCOM AOR versus SOUTHCOM AOR. When you pull that up with MedPros, it's going to tell you the specific vaccines that are required for that location. So you know what you're supposed to give. Acute versus chronic diseases. Those chronic diseases, especially the comorbid ones, usually require vaccines more so. Maybe those that are immunosuppressed, we have to be careful. We can't necessarily give them live vaccines if they're too low. They may not stimulate the antibody response that you really want. We may do travel plans, so travel to endemic areas. If we know they're going someplace, then they're going to probably need a vaccine. You may want to talk to them about it. And health care products. And we'll cover the blood products here in a little bit. Okay, here's your record keeping. Okay. So for this point, beside getting input into MedPros, ASINs, or MERS, right? One of the things that you're going to be able to do as a provider is you're going to determine whether or not you can do exemptions for people, or you can make them permanently immune against some of these things. And say, for example, if I don't have MMR documented in my MedPros, and they look through it and they're like, hey, MMR is a requirement. You don't have it, you need to go get it. Do I need to tell the individual that he needs to go get MMR, knowing that MMR as a little kid is a requirement? Or I can do serology and see if they're positive for MMR, and then I will go into the system and make them permanently immune because of serology testing. So you will have that option and that ability to do. to do that in patients. Okay, so you can send folks out for serology testing, and then they don't have to take it if they've already taken it in the past and it has not been documented. Now understand, as this thing says, there are two types of exemptions here. You have medical exemption, that would be what you do, and then there is an administrative exemption. Keyword there, it says non-medical. What's going to happen is your HR person is going to call you and say, hey, you need to put an exemption in for this person. If it is not a medical exemption, they need to put it in. Because you have two different types. You have your medical and your administrative. If it's administrative, then they have the ability to put in that exemption for you. It doesn't always have to be you medical. All right? Now, that very last one there is a problem, because people always come to us with that. That is not a medical problem. That is not a medical issue. That is a command issue. Any religious exemption will be done through the command, not through medical. Okay? The chaplain will be involved in that. Administrative, medical, lawyers. They may ask your opinion about risks and benefits of the vaccine that they're not wanting to take. But ultimately, it is not your decision. It is a command decision for religious exemption. Okay? And I'm trying to get you involved in that. That is not you medical. Because you're the easy button, which is why they try to involve you. The other big piece there for permanent exemptions, from us putting it in there, if it is a permanent exemption for some allergic reason, they need to have certified allergists agree to that when you put that in there. Okay? That person should go to a board certified allergist so that he can definitely make the exemption approval. Once you get that back from them, then you can just go in the system and just make it done. Okay? But they really should go to the allergist for definitive approval. Okay? So here's a bunch of the acronyms for medical in the system. Okay? Your MI would be medically immune. Right? If it wasn't documented that you did get the vaccine, this is where you can send them off to get serologic testing, and if they come back positive, then you can go in the system and do MI, medically immune. Because you check serologically. Okay? You've got MR, medical reactive, adverse reactions associated with vaccine with benefit risk, does not support continued immunization. Okay? Not give them anymore. MT, medical temporary, pregnant, whatever it may be. Medical permanent. That's some examples there. That's the one that you would wait for the allergist to tell you yes, and you could do MP. And then MD, medical decline, if they have a religious waiver. And if they do decline it, they don't have one, then you just send them off to the command. You don't have to fight with them. Just send them off to the command. Not your choice, not your decision. The administrative folks, when they call you and tell you, hey, you need to put this in, okay? Your answer is, this is an administrative action, not a medical action. These are all the codes that they have: administratively deceased, administratively CE, missing, PCS, right? So the PCS one is a big one, because they'll call you and tell you, this guy needs some stuff. You're like, all right, he needs a PCS, what do you want me to do? Right? They, administratively, can go in there and put this in, because that'll keep them up to 90 days from actually affecting their medical records. Which is really the key point here, is to add all these things there, because you don't want it to affect your medical records. If you've never given vaccines before, this is extremely important. Okay? Rights and medications. Hopefully, for those of you who have, you guys have done a lot of this. A lot of this stuff generally becomes inherent when you do stuff. So So number one is, do you have the right patient? Sounds pretty simple, right? Do you have the right patient? Hey, what's your name? What's your date of birth? That's usually what they do over at the hospital to ensure that you are the correct patient. What drug are you going to give? In this case, what vaccine are you going to give? Okay, where are they going? Why do they need it? So that you can ensure you give them the right vaccine. Next one will be the time. Is it a booster? Is it a second dose, third dose, fourth dose? Okay. Is it the appropriate time to give them the next vaccine? The dosage. Okay. How much you can give them? 0.1, 0.2, 0.3 milliliters, 10 milliliters. How much you can give? Right? Based on age, based on pharmaceutical requirements. Okay. Have to read that stuff. Route. How are you going to give it to them? You give them IM, you give them some Q, you give them the deltoid, you give them the buttocks. It's going to be intranasal, it's going to be oral. How are you going to give it to them? Okay. What about your site? As to location, right? Deltoid, buttocks. And then document. Right? Because if you don't document, it'll never happen. And then they have to go get it again because nobody believes them. And then nobody sends them out for serology. They have to get it again. Right? If you want to try to prevent, you can. Right? If you want to start giving people multiple doses of this stuff, if they don't have to. So the site location, one of the biggest issues that we have is how you give the vaccine. So my suggestion to you, if you are not comfortable or have never done shots before, when you get to phase 2, make sure that you start looking and seeing and practicing there. One of the biggest issues we have, especially when we're going to the deltoid area, the shoulder area for vaccines, is that people do it in the wrong location and they end up going into the joint because they go too high up on the shoulder. Okay? And that is a reportable injury. And there's also a compensable injury, which means that they can get compensated in the civilian sector for that. In the military, that would be good for your retirement, but not good for your career because it's going to hurt for quite a long time. Here's some of the vaccine myths that we're going to have to talk to our parents about or their parents about. Right? Make sure that you give the VIS, vaccine information form. If they do have questions about autism and thimerosal, you can always talk to them and tell them, hey, these vaccines no longer have thimerosal in them. Okay? And if that's what you're using to associate autism with, they no longer carry it. So you just try to give them the best educational information that you possibly can give. You need to become a good communicator when you're engaging with parents, or you're just engaging with patients, period. Right? This isn't about being a good communicator in front of a giant group of people and just giving them information. Okay? That's one thing that you will probably be doing as well. But my point as being a good communicator is you have to express your medical knowledge with your medical information that you give the parents or the patient, right? With empathy and sincerity, so that they truly know that what you're telling them is you're trying to help them the best you can. Some places may have little programs that kind of help you become better communicators with patients. If you find them, and you feel like you got to go, you may go. See if it helps. For those of you in my junior clinical class, I don't know if you've realized, as we talk and I give you an example, my demeanor changes when I give you that example. Right? My body posture changes. My eye contact becomes a lot more direct with the individual patient. have to try to be as clear as possible. And after each statement, again, the way I do it is usually, do you have any questions? I want to make sure that they understand each statement. And that's really dependent on the individual patient, right? You have patients that have barely graduated high school, and you have patients that have their doctorates, okay? So you'll have to address each one depending upon their knowledge level as well. So you may have to break down things really, really low, or you can go ahead and talk at the medical level and be okay with it. So this piece is a little bit difficult with communication. It's an art form that over time you'll start developing as well. So one of the big things that we need to do is give them this information without bias. And I think I mentioned that before. Because it's not for us to make that decision for them. It's for us to give them the best information we can to allow them to make a decision on whether they do or do not want to get the vaccine. This is kind of important. This is what we call the 2-week rule. Okay? If you give antibodies or hemoglobin, okay, too soon after a live vaccine, it can neutralize, okay, the live virus in the vaccine. All right? In which case, your immune system is not going to respond appropriately, so you're not going to develop the antibodies for it. Okay? And that prevents the body from developing long-lasting immunity, which is why you gave the vaccine in the first place. So keep that in mind. If a live vaccine is given first, okay, you need to wait at least 2 weeks before giving any antibody products. If an antibody product is given first, that will be your second line there. Antibody containing product, right, is given first, then you need to wait up to 3 months. Okay. Or maybe more, depending upon what type of live vaccine you gave, right? Because we don't want them to interfere, and we don't want them to make sure that they cancel each other. So number one, live vaccine and antibody containing products go together. And then PPD and live vaccine, those two go together. Okay? So if you're going to give a live vaccine to someone who's going to get a PPD, you read the PPD first, and then you give them the live vaccine. Okay? Or you can give them the live vaccine, wait 4 to 6 weeks, and then give them PPD. And then down at the bottom it says 2 inactivated vaccines. You don't need to wait. You can give them one in one arm, one in the other, or one in one arm, or one in the buttocks. Your choice. Inactivated live vaccines, 1 in 1. Just make sure that you give it to them in different body locations. Okay? Don't give it to them in the same place. Same deal with oral and injectables. Okay? No minimum there. 2 live injected vaccines, none, but given individually, right? You need to wait 4 weeks. Right? Today, if you show up, I can give you 2 live ones, no problem. But if I gave you one yesterday, I'm going to need to wait at least 28 days before I give you a second one. Here's other timings. The biggest questions that we usually get are, hey, I'm past my booster time. Do I need to start the series all over again? If you read most of the inserts, usually you don't. You'll just get the booster and you'll still be okay. Alright? Now, if you follow that link down there, it's going to take you to a site that has all the information about all the vaccines. Okay? I would suggest that you do that later or now, whenever you want, and then just make that a favorite site until you get comfortable with a bunch of vaccines where you can just go in and read all the information that you need for each vaccine. I think I've mentioned to you before, when I was growing up as a PA, I just kept all the inserts from all the vaccines that we did, and they were all stacked on my desk. And as I needed them, I just read through them real quick, highlighted the little areas that were important, i.e. contraindications, right? And dosages and routes. This way I knew how to give it, what to give, and who I could give it to. So you guys will do that however you guys choose to do it, as you continue to grow up in your PA career. Here, contraindications, obviously, if you have anaphylaxis reactions to the medications, and don't give them, right? Makes sense. Causes you encephalopathy in less than 7 days. Prior history, right? I don't give it to them. Okay. Live vaccines for pregnant folks, don't give it to them, right? Unless it's a risk versus benefit thing. If we really need to have a serious discussion. Right? Severely immunocompromised folks, and regardless of whether you give them a vaccine or not, their body's not going to react to it and create those antibodies. Okay, another thing. Precaution-wise, there's a whole bunch of things there to keep an eye on. One of the biggest things that you will see for almost all vaccines as a contraindication is the history of the Guillain-Barré. If you have another slide that has some of that stuff on it, but keep that in mind. Guillain-Barré is one of those like, hey, if you've had it, you may not want to get certain vaccines. And I point that one out because that is a contraindication for influenza. And so if you're going to get influenza and you have history of Guillain-Barré, you probably shouldn't get it. Okay, so here's a whole bunch of other products, like growth medium, egg, protein, influenza, yellow fever. Those are usually grown in an egg protein. So a long time ago, I would say 10 or 15 years ago, okay, if you were allergic to eggs, that was a possible reason for not getting the influenza vaccine. But nowadays, you can read it through the insert, it's been filtered so much of that egg protein that causes the allergic reaction to individuals is almost non-existent. In which case, even if you're allergic to eggs nowadays, you can still get the influenza vaccine. Okay. Stabilizers, MSG, gelatin, you'll find those in some of those vaccines there. Preservatives, as you can see, formaldehyde, phenol, and antibiotics. Yes, antibiotics are used as preservatives and growth in some vaccines because we don't want them to get caught up with bacteria, especially after they've been taken. Adjuvants, we talked about the aluminum. That helps spur your immune system to make better antibodies, stronger antibodies. Now, what we do need to talk about is that interval between blood products, measles and or varicella, right? So, depending upon which type of blood product, right here it's got packed red blood cells, whole blood, plasma platelets, IgG, and Rogan. Depending upon which one, right, these all contain antibodies. So if you're giving them something, they may affect antibodies. Now here you have packed red blood cells. Those packed red blood cells usually have those proteins on the outside portion. If they become washed, if the packed red cells become washed, that means everything's been cleaned off of that protein shell. Then it contains no antibodies to include, which is what causes the decrease of the live vaccine. So if they're washed packed red blood cells, not a problem. You can give it to them. Packed red blood cells are going to wait at least 6 months. We don't want it to interfere with buildup in the system. Because measles and varicella are actually live vaccines, or live attenuated vaccines. Those plasma and platelets, on the other hand, you have to see if those plasma and platelets actually have antibodies in them. If they have antibodies in them, then you need to wait. If they do not, then you just give it to them. It's the antibody piece to all this blood component that is going to cause the interference of the live vaccine in the body developing the antibodies for that. Okay. That intravenous immunoglobulin, okay, if it's collected, okay, some from human plasma, okay, immunoglobulin obviously that contains some antibodies. So again, you have to be careful. That one says 11 months. It could be anywhere between 3 to 11 months for that one specifically. All right. And then you have your ROGAM, which is usually not a serious issue, right? Because mom probably already had the MMR, and if she didn't, she should have had it earlier when she was being taken in as a patient. Right? So usually that's not a problem. Okay? But if MMR is given at the same time ROGAM is given, then 3 months later, or anywhere between 6 and 12 weeks later, you need to do a serology test to verify that it actually works. Right? There may be some interference between the ROGAM and the live MMR vaccine. Okay, so here's ACIP recommendations for pregnant women. Seasonal influenza, Tdap. If you go down to that CDC site there, it gives you specific vaccine pregnancies. Okay, vaccines now recommended for pregnancy. Most of these are live vaccines, which is why you don't give it to them, except for anthrax. There's limited safety data for anthrax, and it's not really one of those common exposure things, so no necessary reason to give pregnant women that. Right? HPV, okay, no benefit during pregnancy, and there's limited safety data for that, so it's kind of contraindicated. Then you have your PVC-13, which is not a routine vaccine. So that would be a risk versus benefit that you would talk to your patient about if it's medically necessary. Otherwise, don't give it to them. All right. So we'll go ahead and stop here for today, and then we'll pick this up tomorrow. Just give major part some time.
So if you haven't already, there are 3 slides posted under the diabetes for the SGLT2 inhibitors. We got excited and then we talked about them, but like not the detailed slides of them. So there's the ones that we are using for our comorbidities.